Good morning, good evening. That is wherever you are or anywhere in this world. Today I am going to speak about the packaging development checkpoints for biologics and biosimilar products. So this is very useful guidelines for those for the specifically for the packaging technologist, polymer science, and MTech packaging, BS packaging, and those are professionals as well. So first of all, that is, so if it is a new molecules, new drug discoveries, as a packaging professional, that is what you have to do, and what should be your checkpoints. First of all, that is you have to take care of, that is first characterization of the product. You have to go through the product characteristics in details, because specifically, unless you don't know the product characteristics, basic things, so you don't know that is in which points, what are the points you have to take care of, like product pH content. Then again, that also, that is toxicology, the toxicology details, also extractable and leachable possibilities for with the primary packaging materials. And you can also see the, that is those products, those molecules, basically products characteristics is affecting the delamination of glass things. These are the things also, that is you have to take care of. First of all, after going through in detail the product characteristics, basic points you have to tick mark and note down in your notebooks. These are the points, that is what I am talking about. So second point is that first you have to procurement of the primary packaging material for the stability studies itself, small quantity. That is for primary packaging material stability studies, that is for 3 months, 6 months, 12 months. So you have to procure according to the sample size. And also, that is after procurement of the primary packaging materials, for example, that is the glass vials or PFAs or cartridge, or any polymeric material, if on the COC, COP, PP vials, composite, that is polymeric, that is glass vials in also COC, COP. So then you have to carry out the stability. You can keep you fill the product, and for the rubber stoppers also you have to use for the rubber plunger for the PFS. So that is you have to, when you fill the product, that is after filling the products, you have to keep the vials in inverted form to make sure that is your, that is product will get in touch with the rubber stoppers. This, that is you can keep, you don't know, for the new drug discoveries, you don't know, you don't have any benchmark samples. For generic product, it is very easier, you know your innovator, so it is very, your life is very easy. So for innovator is for generic product is concerned, innovator, that is sample characterization, that is you have to carry out the, that is vials testings, that is according to that. And you can carry out different types of testings for rubber stoppers. And you can finalize very quickly, that is whether, that is what kind of vials, whether it's uncoated or coated vials, borosilicate, aluminum silicate, or you have to go for the polymeric vials you have to take care of. But for that, so it is easier for generic players. So those who are working in a packaging technologist or polymer science technologist in the respective field and respective projects. So come to the points for the drug discoveries for new molecules. For new molecules is concerned, since you don't know, packaging technology is to be very careful for choosing each and every steps. Like for the sample preparation, that is you have to procure for stability studies, you have to procure borosilicate glass, aluminum silicate glass, coated, uncoated. Coated, if it is coated, that is you have to go for, you have to procure the sample with siliconized, if for PEG or ammonium sulfate flushing inside the surface, So these are the three kinds of samples. See, for the glass vials is concerned, or the similar way, that is you have to proceed for the that is a PFS or cartridges. So you have to take care of the, these are the vials is concerned. And you can, if you go for the polymeric, you have to keep the procure the samples for the COC, COP, PP, or you can constantly, you can three, you can keep total, that is six number of, or six or nine number of the sets of samples that you have to keep. You don't know that is your product is in which packaging material in that is your product will be stable, you don't know. Also that is you will procure the packaging material that time, that is the sterilization, that is packaging technology, so has to be very very careful. That is your primary packaging has to be one set, you have to keep ready for gamma sterilization, ETO sterilization, and steam sterilizations. And similarly for the for the rubber stopper as well. So unless that is you are keep your samples as many as the sample, different types of samples you procure, and keep your product for stability studies, you can save time. And in the you can save time, and in the last moment, that is if your options is less, you don't know, that is your product may be fail, so you will be in trouble. That is you can capture, it will get to capture the market. So your new drug, new molecule is concerned, your activities is tedious, like what I am doing for last 6 years in US. So that your so those are generic in the similar way. That is you have to characterize your innovator samples. Your sample is ready, that is your life is lazy, so you can keep the samples as per the innovator. After characterization, that is you can keep the samples ready, and you can procure the samples for the stability studies. So the second point is that after getting the stability studies result, parallelly what you have to do, for you have to get the extractable studies for, and for the glass vials or polymeric vials from the suppliers, from the vendor, as well as, and they have to specify which pharmacopoeia they use specifically for USP 660, for the rubber stopper, that is 381, and for the extractable and leachable studies, for extract leachable specifically. So few vendors are providing, if it is they provide like BD, so that you can get the their leachable study samples after obviously, that is for leachable studies you have to provide them your product. That is very there should be some agreement signed by you and them for when you will, that is give them your product. So after for that, that is for parallelly, that is you have to go through, if you send your product to US, you have to follow the USP, that USP 660, that is you have to study very thoroughly, and what kind of product, what kind of testing, that is you have to carry out. Everything is in data in my previous that is video, that is I already explained. For the glass vials, that is what are the kind of test you have to carry out. For the polymeric vials or polymeric PFS on polymeric that is cartridges, what kind of necessary testing as per USP, EP, JP, that is you have to follow. So these are the knowledge packaging technologies, that is you have to be always true. Always you have to, this should be the always you have to keep in mind. So these are the testing that you have to do. So analytical people know, but you should know also. That is, so these are the testing that is you have to carry out. For the polymer testing, for identification of plastics, for example, that is analytical people are carrying out the testing for IR, or they are going through the DSC, differential scanning calorimetry. So these are the things that is packaging technologies has to very careful, and they should be knowledgeable, go through the updated specifications. So the when you will that is you get the results from that is final result for on one specific glass vials, for example, that is your packaging material, your product is true, your that is stability study is passed, that time, that is according to that, that is you have to finalize the packaging material specification, glass vial, rubber stopper, then label. I forgot to tell the label plays also the printed label plays a vital role for the extractable studies, for printed ink and coating the wax, that is water based coating or aqua coating, they are and solvent based coating are using. It plays a biggest critical problem they are facing for leachable or leachability, product leachability is concerned. So sometimes this has been observed, that is for those ink are using, inks are using, or also the different types of that is solvent based coating is using, that is it leaches, and also ultimately discoloration observed in the product. So the most important points, these checkpoints are, that is the leachability studies, statistics, or for the label, glass vials labels and labels from the suppliers you have to get, and you have to carry out the leachable studies and extractable studies, especially from the our in-house analytical department. If it is they don't have the facility, you can get it, you can send the material from for outside US FDA accredited lab, get the result and tally, that is okay, your packaging materials, your labels are okay for you, compatible with your product. So you have to keep the three specification after getting the stability result. You have to keep the finalize the specification for glass vials, rubber stopper and labels, bottle labels. These are the very significant things. Clear? I think it is clear. Second step, after finalization of the primary packaging material, parallelly you can go through the secondary packaging material specification, unit, carton, tertiary as well, corrugated box, all these things, that is parallel you have to keep ready, and provide also in between. One more thing I forgot to tell you, that is okay, fine, that is you can provide to the purchase department according to your specification, approved specification signed by packaging development, QC, and the QC and QA people, also the regulatory. These four people will sign on the specifications. Also, that is you have to keep this specification in your file, and after that, that is you have to give this one to the regulatory study for the USFD finding. Next step is after getting getting the specification, that is purchase department will procure the primary packaging material. After that, after receiving the primary packaging material for commercial batch, QC also parallelly you have to provide the approved specification to the QC people. Those people, that is they will carry out the different kind of testing for the primary packaging material as per the equal studies. Mostly that is sometimes packaging development people is preparing this one for equal chart, that is all the what are the parameters and which one you have to accept, which one not. Packaging development department and QC will sit together, and it is better always, and to then finalize the packaging material specification. When you finalize this packaging material specification, along with that, that is the the equal also should be there for the primary packaging material. This is easier for them, this has to be signed also, from the respective department, packaging, QC, QA and RA. So according to the, that is by seeing, but your equal has to be with photographs. For example, while rubber stopper, all the photographs should be there. For example, cracked, dented, which one that is approved, that is, if you see the dent, if you observe, you crack, if you observe along the circumference of the rubber stoppers, leak test will fail. That is when it will not compatible with the when you will put in the that is vials, that is your flipping, that is you will observe, and your after aluminum foil, aluminum seal, even though that is your leak test will fail. So the packaging technologies has to be very very careful. The quality different, all kinds of what is the quality studies and what are the parameters that is QC will check. Packaging development department has to be thoroughly knowledgeable about wide range of defects, different foreign particle inside the, that is in the rubber stoppers, that is excess material, that is material, and also that is it is observed, that is here you can observe sometimes that is you have to carry out the moisture content WBT here. So there are many others, all these things. Parallelly, that is for the glass vials also, that is what kind of AQL, what are the different parameters with photographs, that is the in AQL should be there. So whether it is crack, foreign particles, bubble, that is in the surface body, of the surface of the, if it is blowback vials and blowback rubber stoppers, they have to check the compatibility, put the rubber stoppers and check also. After getting, after finalizing, that is when the QC will pass the packaging material, primary packaging material as per the AQL. Then they will release for the commercial production. When they release the commercial production, when the commercial batch is ready, before that stability study batch, one more thing, three stability study batch, that is that is batch, that is you have to keep ready. That is after that, that is normally the commercial batches is there. Also that is these the when I'm talking about come to the commercial batch, that is when the commercial batch material is released for the commercial batch. What during, before start the commercial batch, what are the action that is as a packaging technologies will take care, what should be the checkpoints. So in the packaging line, that is they have to see that is whether if it is rubber stopper, for example, if it is igloo type, if it is the lyophilized product, that is if it is igloo type, that in different types of live stoppers are available. If it is igloo type, single slotted or double slotted, that is when before that, that is the everything is finalized before it is come. If it is igloo type, that is during filling of the product, that is vials, for example, we have to, in the vial itself we have to see that is whether two rubber stoppers are sticking together, and the placement of the rubber stopper before closing, that it is placement is suitable or not. After closing the rubber stopper, we have to check in the online, that is whether it is bounce, rubber stopper is bounce back or not. We have to see. And also that is when it is sealed, that is with the aluminum foil, that is seal, that is whether sealing is perfect or not. After that, after getting the that is the vials also, that is we have to take care for the leak test. The leak test we have to carry out parallelly on the online, you can say. Also the when after filling the bottles, that is vials, that is we have to apply for labeling section. We have to take care of the positioning of the label, batch coding, barcoding instructions. These are the things, the when you will specify the complete the packaging material specification. And parallelly, that is in the label printing will go, and unit carton artworks preparation you are talking about. Before commercialization, I forgot to tell, before commercialization, because packaging material specification, when you will finalize, parallelly, that is you have to finalize the artwork by consulting with BD, RA, QC. These are the people, and the QC, these are the department, that is you have to take the signature of the after preparation of the artwork. This artwork, it has to go, after signing, it has to go to the printer, and they will send for the send to the supply to the that is production line. After that, that is QC will check according to that. So this is the packaging development. So packaging development responsibility from development R&D, packaging development R&D, still from stability batch study, still three commercial batches. The three commercial batches successfully complete the three commercial batch. by monitoring, that is packaging development department has to monitor, then their responsibility is complete. Not like packaging development department is only involvement in R&D activities. Forget it. You should always remember packaging development professionals responsibility from R&D to till the completion, successfully completion of 3 commercial batches. So hope. Then logistics and distribution, that is there are few companies, they involve the packaging development department for what kind of pallets, that is definitely packaging pallets they have to that you can prepare the specification for the pallets, palletization, how it is stretch wrapping. After palletization, how many shippers will come, what should be the height of the their individual pallet. So these are the things is that is international, that is palletization, that is you can see the specification, you can standardization for USA is one and US, that is US, Europe one, and JP, that is you will get in different pallet size and palletization, that is high. After that is, and what kind of in one container, whether to 20 feet or 40 feet container, that is how many pallet will go, that is so that is you can take care of. But these logistics and distribution part, there are many companies are separate department, they are warehouse logistics department, they are taking care of. But as a packaging technologist in US, I am talking about our responsibility as a packaging development. They are actually there is no that is position like that in the packaging development in US. Basically this is since I am in the new drug discoveries, so I am in the new drug discovery project in the device innovation, like that is our position. Principal consultant, principal guy for the device innovation, like that. There is no in US like packaging development. It is in Asian country, it is, yes. So there are few companies like, yes, those they are specifying like packaging consultant, like the packaging development, like that. So I think that is, as a packaging technologist, what that is, it will be very helpful this video. Please take care of. So life is not so easy, that is when you will be work in the new drug discoveries project, and there are a lot of things that is you have to take care of. You have to be thorough knowledge about the different pharmacopoeias and logistics and distribution studies, container specification, palletization specification, and different rules and regulation about labeling barcoding systems, and barcode serialization systems for primary packaging, secondary packaging, tertiary packaging, how you are doing. That is, I'm talking about these one also, the packaging professional responsibility, also selection of their companies that they are involving, that is packaging technologies for selection of barcoding serialization machineries, and labeling machineries, and the automations also, that is. So I am that is also, that is I have a vast knowledge about the procurement of the packaging procurement, FAT. They are involving the packaging technologies for the FAT. I have the very good knowledge about the different kinds of that is machines with artificial intelligence, robotic arms using. So there are lots to do. So basically these are the things our packaging technology has to take care of when they are involved in the production of the 3 commercial batch. So packaging technology has to be very careful and thorough knowledge about the commercial machines also. Be careful. So you have to be very knowledgeable and take care of all these things. I think if you follow this guideline, that is, you will not miss any points. So you will not never get any market complaint. So thank you very much for seeing the video. Hopefully that is tomorrow I will come up with new video. Good morning, good night. Thank you very much.